good evening, everyone. My name is Jorge Bogantes. I'm the Natural Resources Specialist at the Anacostia Watershed Society. Can you hear me well? I guess so.
Yeah, I hear you well.
Yeah, so yeah, I'm gonna talk about Mussel Power for a healthier Anacostia River. So why mussels? We started finding mussels in the river because we do a lot of wetland work. So we started finding shells and live mussels like this one. And this one was funny because I was fishing with a friend, and when he was reeling in at Bladensburg Waterfront Park, this mussel snapped onto the line. I had no idea what it was. I was sure it was a mussel, but I didn't know much about mussels by then. So I sent a picture to the Maryland expert, and he identified it as eastern floater. So eastern floaters happen to be one of the most common mussels in the East Coast, let's say, and other parts. So the first thing that everyone asks is, can you eat them? Uh, the short answer would be yes, but you don't want to eat them. Why? Because they are said to be not very tasty. They taste like mud. They are not like the ones in the picture, which are marine mussels that are way tastier. So, and of course, you don't want to eat them from the Anacostia River because of the pollution issues that we have. And I'll talk more about that. So, Native Americans used to eat them, but they were mostly used for the shells, for ornaments and such. And then in the colonial times to make shirt buttons until relatively recent when it was replaced by plastic. All right, so why mussels in the Anacostia River? Why now? Well, we have this huge project in DC. Is the biggest infrastructure project since Metro. Is the Clean River project, and these tunnels. This was during construction. This is part of the Anacostia River Tunnel, which is now working and is capturing 88% of sewage. So is a little bit overperforming. And in the process, it also collects a lot of storm water and tons, thousands of tons of trash when it rains. So that's a huge milestone for the Anacostia River. Actually, AWS filed a consent decree back in the day in the early 90s to make this happen. And, it took all these years, and DC Water, which used to be DC Water and Sewer Authority, that was the former name, is now one of our main partners. The Anacostia River has been improving. As you can see in our State of the River report card, and got a passing grade. Might sound mediocre, but it's actually a huge step forward. It's the second time it gets passing grade. And as you can see, the variables and the green arrows, it's been on an improving pattern. There's only two variables that have not been improving: dissolved oxygen and stormwater runoff. There's still, we live in an urban area, so there's a lot of impervious surfaces, a lot of stormwater runoff. Is still an ongoing issue. But the fact that water clarity and other variables are improving makes it a good time to do this. And then we met the Virginia Fisheries and Aquatic Wildlife Center, also known as the Harrison Lake National Fish Hatchery in Charles City, Virginia. That's southeast from Richmond. And they are the biggest mussel hatchery in the greater Mid-Atlantic / Southeast region, run by the Fish and Wildlife Service and the Virginia DWR. They really have made this project possible because they are the experts in propagation. They have indoor and outdoor facilities, and they produce the juvenile mussels for us. Why mussels? Again, because they are filter feeders. Mussels eat stuff from the water column, which can be E. coli, which can make us sick, protozoans, a lot of plankton, and other types of microorganisms. I was going to add one joke for Serenella, because they do also eat spores of aquatic mushrooms. They are known to eat spores from aquatic mushrooms. I didn't get a good picture for that, so I was gonna put a picture of shiitake on a soup to depict it. But in this demonstration, what you can see is a couple of 5-gallon tanks. The one on the left has about 11 adult mussels. The one on the right doesn't have any mussels. So the picture on top is the before. The picture on the bottom is an hour after. So you can see the water clarity difference. You can even see through on the bottom picture. You can see the rocks on the background. So one adult mussel like this can filter between 10 and 20 gallons of water a day. It depends on the species, the gill morphology, so they have different rates, but that's kind of the range. So let's go back to the bivalves. So mussels are bivalves. They are mollusks. They are related to snails and slugs, but more closely related to the bivalve. They are bivalves because they have 2 valves, 2 shells. They are related to scallops, clams, and others. There's over 10,000 species of bivalves in the world, a lot in the oceans and brackish waters. They are present from the Mariana Trench, the deepest part of the ocean, to our local vernal pools with the fingernail clams. They've been here for a long time, over 500 million years. And interestingly, there was a period that is called like the Permian extinction period, which was the greatest mass extinction, that saw the greatest mass extinction in Earth's history, about 250 million years ago, because of severe climate change, global warming, and a lot of CO2. It wiped 70% of life, but only bivalves and snails survived pretty much. So some interesting facts. Another one that you may like, the picture on the right depicts a clam with the adductor muscles. Now, these are not the animals, these are the tissues. So these tissues, that they are cylindrical in shape, they allow the animal to open and close the valves. So on the picture on the left, see the yellow arrow, that's a scallop. That's when you eat scallops, you're eating the adductor muscle, the tissue that allows the scallop to open and shut. These, although we're dealing with the different kind of bivalves, these guys are freshwater bivalves, freshwater mussels. They belong to the Unionidae family. There's over 1,000 species in the world, but the US is really the hotspot. Especially the South and the Southeast, South is a world hotspot of unionid freshwater mussels in the world. They are actually the most imperiled animal out there. 70% of the North American species are imperiled due to many things. Pollution, damming of the rivers. Back in the day, they were over-collected to make shirt buttons. So many threats to them. So 70% imperiled. If you compare to other animals like birds or mammals, it's about, 10, 15%. So they are really, really in danger. And Alabama has the most species with 180 species. Maryland has 16. DC around the same, including the Potomac and Anacostia waters. Virginia has more, like 85, but that's because of the Clinch River and Tennessee River. All those drainages there in southwest Virginia are super diverse. So they have that plus the Atlantic drainage species. Now, this is the fun part about mussels. The life cycle is super fascinating. So freshwater mussels depend on fish to complete their life cycle. Uh, on this diagram, this is an eastern pond mussel. So first, the male release the sperm into the water column. Female intercept the sperm and then fertilize the eggs that are in her gills. And then the eggs turn into larvae inside her gills, in pouches inside her gills. And that larva is called Glaucidia. You can see on my background and also on that photo, they are microscopic and they look like tiny mean Pac-Mans. They have two valves, just like their parents, but they have hooks at the end of the valves. And those hooks allow them to attach into the fish's body. This is their parasitic life stage. That's when they eat bodily fluids and tissue from their host fish, which can be a very specific species of fish or can be different species of fish. In this case, the eastern pond mussel uses the white perch, or it can use largemouth bass. A lot of the Anacostia species tend to be on the generalist side. They can use different fish, although for hatchery propagation, they normally use one that is like the tried and true fish host. So after that, so this parasitic stage lasts about 3 weeks or a month. And after that, these larvae drop down into the riverbed and start growing as filter feeders. First, it's funny because they use their foot that looks like a tongue to grab particles, but soon after they become full filter feeders as we know them. And they can be as tiny as a fingernail in the picture, and they just keep growing and growing, as you will see. And this is where I want to show you something. This is, so mussels go even further to attract their host fish. And the lamp mussels, lamp as you know, light lamp, that's because they have rays of green color. Maybe that's why they put them lamp. So the lamp mussels attract their host fish in a very interesting way. Let me show a video right here. I'm gonna stop the share and go for the YouTube video. All right, hopefully everybody can see it right now. So this is an eastern lamp mussel that I taped underwater, videotaped at Buzzer Point at the confluence with the Potomac River. So as you can see the fake eyes? And see how the female wiggles the lure? That's to attract the host fish. And the fake eyes make her look like a minnow. And that's the point. That's to attract the host fish. So she starts wiggling the lure. The fish, in this case, like a largemouth bass or a bluegill, will come and bite, and then she will release the larva, and the larva attach into the fish's body. That's when the parasitic stage starts. Let me see. Oops, it's not changing into the next slide. Maybe now. Okay, right now. So I can hear some music there. Maybe it's someone.
I think it's actually the next video on the YouTube might be playing automatically from what do you had up?
Oh, yeah. Let me close it. Sorry.
That's
Sorry
okay.
about that. Okay.
And once you get the presentation back up, we actually have a couple questions.
Okay, let me open the presentation again. And can we, what are the questions?
So our first question is, how does the larvae get released from the host?
Yeah, they just drop down. They just, sometimes they become like encysted in the tissue. So they just manage to get off from the fish body and then they drop down into on the riverbed.
And then do the host fish die after the larvae invade them?
Not necessarily, not normally. It's a parasitism relationship. Only in very extreme cases, let's say if a young fish gets like a cloud of larvae in the gills, that might compromise the fish, but normally it's not a problem. It's just like having lice.
All right. No, that's all the questions for now. Oh,
So, oh, that's the food on the picture. See, you see like the white tongue-like appendage. That's the food that they use for locomotion and to bury themselves into the substrate, in the mud, sand, or gravel. So we've been doing surveys since 2015 in partnership with the Maryland DNR, and then we continue doing our own surveys along the tidal and other adjacent non-tidal areas. So we found that Buzzard Point, right by the confluence with the Potomac, is a hotspot. You know, you've got a real mussel bed with hundreds or even thousands of mussels there. Um, that's James Creek Marina on the background. And then the main stem of the river was rather poor for obvious reasons like historical pollution, sedimentation, and such. But the adjacent wetland systems like Kingman Lake and Kenilworth Lake are working as sort of the refugia. There were, there were decent diversity of species. Numbers, yeah, it was more patchy. One mussel here, one mussel there, not quite a mussel bed, but mussels were, hanging in there. So that's why we call it the mussel refugia. And these places are tidal impoundments. They are not actual lakes. They are tidal impoundments connected with the river hydrology. So we have found a total of 8 native species. SGCN means species of greatest conservation need, either in Maryland or DC. So these are the species we're working with so far. LY floater, eastern lamb mussel, eastern pond mussel, and more recently we added the eastern floater. Actually, this morning, I found 2 gravid females, and I'll talk about that. So these are the 4 species we're working with. So the first phase of the restoration project started in August of 2018. So we put mussels in floating baskets, and the idea was we monitor them every month to check for growth, survival, and the site suitability. Because they were floating, we were kind of testing how good the water conditions are in terms of how much food they have and such. And with this, we also helping with the conservation of some of these species that are listed as species of greatest conservation need. And coming up, we still don't have any results, but we will have some tissue analysis done, checking for nutrients, nitrogen, phosphorus, and microplastics. The idea of the nutrients is to make mussels a best management practice for the management of these nutrients and sediment in the Chesapeake Bay region. Other people are working on this goal as well. So the idea is the more mussels, the more biofiltration. So the process starts with me swimming, snorkeling in the Anacostia River at Buzzard Point. Only in the other places I don't snorkel, only at Buzzard Point. And so we find the gravid females. What does that mean? That's the pregnant females. How do I tell that? So I use some repurposed nasal speculums to carefully pry them open and check for the gills. If you see the yellow arrows, they are pointing the gills of a gravid female. They are swollen, or they look swollen or ribbed. If a mussel was a male or a young non-gravid female, they would look like deflated, like a deflated balloon. So that's the first process. So we send those females to the hatchery in Charles City, Virginia, and then they take care of the nitty gritty. So they do the host fish infestation on indoor tanks. They collect the tiny larvae, feed them at different stages, a cocktail of plankton. And when they reach fourth of an inch, they put them in ponds out there. And then once they, they reach about, half an inch or so, they are ready to be released or to be put in other baskets and cages like we did. And those females, they get tagged and then they get released. They live. That doesn't kill them. So we release them back into their place where we collected them, and they keep living. So these are the sites, the 7 sites or 8 sites where we started doing our project. Some were kind of deep waters on floating docks, or others shallow waters fenced in this kind of fencing. Many thanks to a lot of the people that have helped, including perhaps some here in the audience. I know for sure Bradley has helped me out there, and Friends of Kingman and Heritage Island, Environmental Systematics help us a little bit with data collection, Master Naturalists, Watershed Stewards, and just volunteers, either independent volunteers or from companies like FedEx, our awesome interns. Many people have helped. So mussels grow their shell outward. So if you see this part that looks pale is the oldest part of the shell. So when we got the mussels, you see this line of growth, they were this small, like this kind of brown, light brown color. Now this growth that is pointed by the arrows is the growth they gained in a few months growing in baskets in the river. You can see the nice green hue of color. Young mussels are very pretty. As they age, they become darker, depending on the water conditions, but that's normally the rule. They become darker, but when they are young, they tend to be very colorful, and different species have different colors. This is an LY floater, the same species as the previous slide. The one on the right is a young one, a juvenile from the hatchery, and the one on the right is a the full-grown shell of, it was a dead animal. It's just the shell, but that's what it looks like when it reaches complete maturity. In this case, these guys can live for 20 to 30 plus years. Now, one of the species we have, the Eastern Elliptio, which also uses the American eel, is the most specific one, uses the American eel as host fish. That species lives up to 100 years, so that's pretty amazing. So this is a box and whisker plot of the shell length in millimeters, medians of shell length. So this is when we put the mussels in the river in August of 2018. So you can see the growth is pretty uniform. It was around, upper 20s. It was around half an inch. And then a year later, in August of 2019, you can see the difference in growth. So the top 3 sites were at Kingman Island, the 2 Kingman Island and Kingman Marsh, and the Yards Marina. Those were some of the top, and also Kenilworth Lake, the areas that I mentioned that we still have that refugia for the wild mussels. So they had an amazing growth, as you can see in this picture, in a more visual way to show it. On top is a pond mussel, and on the bottom an LY floater compared to a quarter coin. So they went from about an inch to 2.3 inches a year later. So growths of 90%, and LY floater an impressive 154%. Now, Eastern lamb mussels, and not as fast. They went in a year 29 to 52%, but that's a known. That species is known to grow slower. Do we have any questions?
Yeah, I was actually just about to ask. So the first one is, how do you count the age? Is it the rings on the mussels?
Well, in this case, we know because they came from the hatchery. Now, but yes, you can age an animal, a mussel, by doing a cross section and looking at the growth rings, just like a tree. Um, Actually, if I go back, let me see the growth ring here. This growth ring that is very marked marks the end of a growing season, and then the rest of the growth is attained during the spring to the fall.
Okay. And then we have two others real quick, and we were talking a lot about data, and someone's wondering who did the analysis for microplastics.
For microplastics, we have still in process Dr. Lance Younkus from the University of Maryland Laboratory of Science and Technology. And for the nutrients, we're going to have the Chesapeake Biological Laboratory, which is part of the University of Maryland Environmental Science Center.
Okay. And then just one more. We had a question about how do snakeheads affect your mussel restoration?
We don't know any ways that snakeheads affect them, because snakeheads tend to be more piscivorous. They tend to eat more fish, and they might eat some crayfish, but they are not known to eat at least a lot of bivalves. And what actually could be interesting is if they have become a host, because generally species like eastern floater that go for a wide range of host fish, they might be using snakehead as a host. But again, that's speculation.
Do you
don't
think
know.
there's any worry of snakeheads predating on the host fish and having those drop severely?
Well, that could affect for sure. That could be a way where they can affect. Now, I'm more concerned right now about catfish, especially blue catfish. Blue catfish are native to the Mississippi drainage. They were introduced here many years ago in the Potomac Basin, and catfish eat everything, everything that is organic in front of them, they will eat it.
Wow.
They are known to eat mussels. So yeah, that's my biggest concern fish-wise.
Right. That was actually a question just came in asking what species typically eat the mussels. So other than catfish, are there any others native to this area that eat them?
Yeah, well, we have the brown bullheads, which are native catfish. They are smaller. They may eat some. Also the eel, interestingly, so like I mentioned, the eastern elliptium mussel uses the eel as a host fish. One of the theories for this relationship is that the eel tries to eat the mussel, but if it's an adult, they are kind of like big, and so it's hard for them to swallow, and then they eject them. Unless they are really small, they can swallow them. But during that attempt of predation process, the female mussel releases the larva, and again, eastern elliptium is the most interesting because it only or mostly uses American eel as a host fish. Is the most specialist of our mussels in terms of the host fish relationship.
All right, very interesting. That is all the questions we have for now. I'll let you know when we have others.
So we've had great survival, 92% survival in at least during that first phase. Again, the Yards Marina and Kingman Lake sites had the highest survival. And the in terms of mortality, the pond, we had mussels at the Kenilworth Aquatic Gardens, which I'm sure a lot of you know, had a high mortality. As you can see in the table, it was 45%. Why? Well, it happens that, these ponds are closed systems and they are full of muck and plants, from hydrilla and many kinds of submerged aquatic plants to emergence and floating wetland plants. So these plants consume a lot of oxygen. For example, like the hydrilla and the underwater grasses, during the day, they produce oxygen. And at night, through respiration, they actually consume the oxygen. So in summer and in the early fall, still when it's warm, they consume a lot of oxygen because they are at peak of their growth, and they start prepping for the winter, and they are also, propagating themselves. So anyway, they consume a lot of oxygen, so the dissolved oxygen levels just plummet. And in that pond, it was around 0.5 milligrams per liter. That's super low. Now, the Anacostia River, tidal Anacostia River, averages 3 milligrams per liter, which, all things considered, is actually low as well. But mussels seem to be fine with that. Now, when you get like 5 milligrams per liter or above, that's pretty good. Like, most aquatic critters live happily in 5 or more milligrams per liter. And that's one of the variables that I mentioned at the beginning, that it's on the decline, the dissolved oxygen in the river. Why? We don't know why, but so far it's still in the 3 milligrams per liter. Mussels are doing fine, but we don't know why it has been declining in the river. One of the theories is that because of the tunnel that I showed, there's less combined sewer overflow, which had a lot of storm water, sewage, so that kind of churned the water, and in the process bring in more air and oxygen into the water. With less overflow events during rains, there's less churning of the water. But again, we're actually reaching out to experts to trying to figure out why is this decline in the dissolved oxygen. Now, in a stream, like the Rock Creek or any other free-flowing, high-energy stream, you're gonna get high oxygen, 5 or more. And now, during the cold season, the oxygen also goes up because most of the living things are shut down, sleeping. So anyway, we had to put a bubbler, an aquatic aquaculture grade bubbler, to keep the mussels, and that's why they do at the hatchery to keep the mussels at Kenilworth Aquatic Gardens. So also during that year, the 2018, that was the wettest year on record in our region, 66 inches. That's a lot of water. And so mussels didn't seem to be bothered by that. They had great growth, great survival, and even in that year, there was a dissolved oxygen improvement and chlorophyll A improvement. But again, the dissolved oxygen in the last few years has been declining a little bit steadily. Now, fecal bacteria, there's no shortage of that. Even now, even without the tunnel, there's still some, but you have to consider that it does not come only from sewage. It also comes from wildlife and animals and dogs, dog poop. But, that's mussel food. Mussels eat E. coli and other bacteria. That's one of the reasons why you don't want to eat them. Second phase from October 2019, last fall, to now. So we have funding from the District Department of Energy and Environment, DC Water. So we're doing more floating baskets, this time a couple of new sites, but also with a more community science approach. We have people helping us monitor, and we're educating the public about mussels and their ecosystem services and benefits. And but we also have bottom cages that you see on the right. These are cinder blocks with a lid that is made out of rebar and crab cage mesh. And I'll talk more about those. So those bottom cages are new. They have never been used. They were designed by the hatchery partners. It's the first time they are ever used. And even though we have had some issues, there's also a good side. And I'll mention that. So first, the issue is sedimentation, especially at places like tidal Nash Run. We get a lot of sedimentation, even it's silt up even to the top of the cinder block. Also mussels, some mussels escape because these are not 100% enclosed like the baskets. You know, between the cinder blocks, they can crawl out, or from above if it's silted up. There seems to be competition with Asian clams. On the photos of the right, this was at Nash Run by the Kenilworth Aquatic Gardens in the tidal area. So this is where Nash Run meets Kenilworth Lake, the tidal impoundment. So just in one cage, we pulled out all those clams, Asian clams, on the top. There, as you can see, clams are more roundish, and on the bottom, just a handful of mussels that we had deployed. So yeah, we think there's some competition issues going on with the clams. At other sites, it wasn't as bad, but we still find a lot of clams because they are very abundant in the river. And when we do mussel surveys, we find really more biomass of clams than mussels. Also, it can be harder to find the sample because mussels actually move a lot and they bury into the substrate. So when we're finding for our sample, is sometimes we don't find the number we want. Whereas in the baskets, you know, you have full control and you can just rinse them and get the mussels and then count them and measure them and put them back in. But on the bright side, these cages are very interesting because the mussels have been growing more, at least as our preliminary data tells us. The average increase in shell length in 10 months has been greater than that of in the baskets, the growth they had in floating baskets. So LY floaters, you know, it went from pond mussels 129% and LY floaters to as much as 139%. And this is in 10 months, not even quite a year. So they are growing more. It makes sense because these cages are on the river bed. That's where mussels are. That's their microhabitat, and the density, because in the baskets we put 250 to 350 animals, so they are kind of packed. There's a bit of an overpopulation, whereas in the bottom cages there's less density, so there's less competition between them. Let's see, there's some other questions.
Yeah, there was one question about how do the tags stay on the mussels? Are they stickers?
The tags are little plastic tags that we purchased from a vendor in Australia, Halprint, and they are pasted with crazy glue. They last for a long time, yeah, for many years. If pasted properly, they can last for many years. Yeah. Yep.
The question I guess I have is there any detriment to using the crazy glue on the mussels?
No, once it dries, it's pretty much inactive, and we haven't seen any issues, nor do I have read any articles of people having issues with it. Yeah, go ahead.
Sorry, we also just had one more question come in, just asking if they filter their entire lives. Are they continuing filtering throughout their lifespan? Okay.
Yeah, they from that moment when they drop from the host fish, they become filter feeders until they die. Yeah, they keep filtering, and the bigger they are, the more they filter. So we keep them in the cages, either the floating baskets or the bottom cages, for a year at least. And then after that, we release them into the river. Now, as some of you may have heard, there's a process going on to clean the Anacostia River. There's been a lot of studies to clean the toxic sites and remediate the sediment and at the bottom of the river. There's been a lot of studies over the years, and now we're reaching the point to where they're gonna do the actual remediation work, which is gonna entail dredging and or capping with sand or other materials like biochar that absorbs pollutants and such. So that's why we're only releasing animals at two sites, Kingman Lake and Kenilworth Lake. Actually, Kenilworth won't be affected by remediation, but Kingman will. So we plan to do a salvage at some point before remediation. But so far, we have released over 17,000 mussels between last fall and this fall. We released some this week at the restore section of Nash Run in DC, at a site where DOE did some restoration in the stream. And that was our first non-tidal, because Kenilworth and Kingman are tidal river sites. So just in 2019, we released 7,700 mussels. This year, we have released 9,800 mussels. So if you think about the biofiltration, it can be pretty amazing. And this was just an article at the Washington Post, and when we had a reporter during one of the release events at Kingman Lake. But if you consider the biofiltration of those 17,000 plus mussels at 10 gallons of water a day, that's 64 million gallons of water in a year. That's the equivalent of 97 Olympic-sized swimming pools during a year. So that can be pretty amazing. And that's one of the reasons we are doing this project, because of the biofiltration benefits of mussels. But not only that, they improve the ecosystem, the aquatic ecosystem, for other species. You know, even when they are dead, their shells are used as a refuge for crayfish, mattoms, the tiny catfish, and some fish like mummy chugs lay their eggs on the half shells. So even after they die, they are very helpful for the aquatic ecosystem. So we monitor them. We have been monitoring them even before the release in 2017. But because we have tagged animals with the Halprint tag that I mentioned on the right, you can see it. It has a letter and a number. That's the code. And actually that mussel on the picture, the A224, is a lamb mussel that we found this year at Kingman Lake. So it was released last fall, and it had grown 10% since last year, since it was last measured. So I was super excited to find that was the first animal we found again post-release. And we're doing surveys. Actually, tomorrow I'm going to be doing a survey at that same area. We might find A224 again. And other method that we're using is the PIT tags. It's like a nice technology with radio signals. It's called the passive integrated transponders. And so we put this little thing right here on the shell that looks like a Duracell, is enclosed in glass. And then we put, again, crazy glue to cover it. And then there's like a detector, looks like a metal detector. Then we come in back, and then we can find them if they are at a reachable location with the radio signals. So this is great because it tells us a lot of information about movement of the animals. And the same as the Halprint tag, it tells us growth, survival. Is very helpful for the post-release monitoring. And so one of the other things we're doing is education, not only through the volunteering, but also educating kids. We're showing kids that we don't have oysters, because oysters live in brackish water. We have mussels, and more than one species. We have 8 species working hard with the biofiltration, helping us clean the river. So because of that, we started working on a program. It's called the Mussel Power program. It was modeled after the Ohio River Foundation program, and we started it in the winter of 2019. And it's a 3-part education program, just like we do for our other programs. So it has the classroom session, field experience, and boat tour in the Anacostia River. Well, actually, this is an old number. We have engaged like over 500 students during the first year. This year it was very challenging because of the pandemic. We had to get all the mussels and from the classroom before they died, or because no one was at the schools in the spring. And we were using eastern pond mussels, but also lamb mussels, because they tend to be more resistant of low oxygen. So in case the bubbler stops, if there's a blackout, those species have more chances of surviving in the classroom without the aeration. This is a cute painting by a student, Daycare by Connor. So you can see, it says, a fish is like a daycare for Glaucidia, and you can see the gills like lines, and then the Glaucidia or the larvae look like triangles. That was an awesome drawing by one of the kids. And they also wrote poems about it. We have a little book about it that is awesome. We're also engaging high school students from the Prince George's County system, including from underserved communities, students that are interested in science. So it's been a great engagement process with these students. And again, we have funding from the district government. So we're propagating a total of 35,000 mussels in this second phase. 5 species. Again, I mentioned Eastern floater, we're adding it. That's the new addition. So far, we have the juveniles at the hatchery. We haven't released any of those yet because it's kind of a new species. And then next year, we want to use Eastern Elliptio. That's the one that uses American eel. But the cool thing is that we're going to be, the hatchery folks are going to be propagating Eastern Elliptios in vitro, because American eel is a hard fish to keep in tanks. So the experts have found a way to forego the fish and do the process, the larva growth in vitro. So that's pretty cool. And they did their first trial this year and had a lot of success. So we hope to do that with mussels from the Anacostia next year. We're also crafting a mussel restoration plan. And again, we're gonna have some lab analysis to show like nitrogen, phosphorus in the tissues of mussels to help with the future BMP, best management practice for nutrients and sediment in the Chesapeake Bay. Other folks are doing this work, so this will just amount to the evidence in that regard. And the Anacostia River that you see, the Anacostia Watershed on this slide, is 176 square miles. We have only worked on the tidal river mostly, is where you see the red line, the red arrows. The next frontier will be the tributaries. We have a lot of, hundreds of miles of tributaries. What's the issue? Stormwater. At the beginning of the scorecard of the river scorecard, I show you that 2 variables were not improving: dissolved oxygen and stormwater is also one of them, because we have a lot of impervious surfaces, a lot of high volumes of stormwater. So when it rains, these creeks and streams are super flashy, and mussels like stability. If there's a lot of flashiness, they don't survive. They just get washed downstream. That picture on the right is near my house, northwest branch, lower northwest branch of the Anacostia River in Hyattsville. During a 2-inch rain event, I mean, that's the energy and the volume of water is sheer there. So So yeah, I would like to thank our donors and volunteers. And if you have any other questions, I would be happy to answer them now.
All right. We did actually just get one coming in the chat as you're finishing, and it's asking, like, I guess how much on average are the number of larvae on the host fish?
Yeah, it can be thousands depending on the size of the female. For example, one eastern lamb mussel at the beginning got like 48,000 Glochidia. I've heard from the hatchery they collect huge LY floaters from the Rappahannock River in Virginia, and some of those big ones can have over 1 million Glochidia. Now, not all make it. There's mortality during the first couple of months because they are super tiny and fragile. Once the bigger they are, the more chances they have of survival. Yeah. So, yep, go ahead.
Sorry, we just had a couple other questions come in. Are you done answering the first one though?
Yeah, what's the other question?
So the next question is, will the mussel population be able to repopulate itself in the future without us having to be involved?
Yes, that's the idea. So what we're doing here is augmentation. One of the good things about the Anacostia is even before we started this project, we saw signs of reproduction. We found gravid females, pregnant females, and we found juveniles, which is a good sign. In other parts of the country, like in Oregon, there's a river, I forgot the name, I think it's part of the Columbia River Basin. They found a nice mussel bed with lots of adults, but there's no signs of reproduction. And that can be because different issues, disease, climate change, and many things. So we're lucky that, in the Anacostia River, after all this, history of pollution and neglect, these mussels made it, and they are resilient and they are reproducing. What we're doing is increasing the numbers. Like I told you, like Buzzard Point, we found that Buzzard Point is the hotspot. There's a lot of mussels there. The more we went upriver, the less mussels we find. So what we wanted to change that. We wanted to put more mussels in those areas so that they, from there on, they do their thing, multiplying.
All right. And then the last question in the chat for now is, what is the jurisdiction of the Anacostia River? So, like, who owns them / who is responsible for it?
Yeah, the Anacostia River is very interesting. Well, first of all, the watershed, 85% of the watershed is in Maryland, in Montgomery County and Prince George's County. The rest 15% is in DC, the eastern half of DC. And sorry, that was my daughter, Aurora. So the most of the riverbed is owned by the National Park Service. That's in the DC area. Yeah.
Well, it's nice to see your daughter too. We did get 2 more questions come in as you're answering that one. When you're surveying, do mussels use their foot to get away sometimes?
Not really. Well, the first thing they do, they just shut, they close. That's the self-preservation instinct, is just to shut. Yeah.
And then the last question for now is, how do you expect climate change to impact mussels in the U.S. and around the world?
Yeah, that's a very interesting question, and I'll give you one anecdote we had this year. So eastern floater is the new species we have added into the mix of propagation. When I was collecting the brood stock, the gravid females in the spring, so this spring started very warm, but it got very cold in about April. What happens is that the species that release the larva in the early spring, they were gravid ahead of time. So when I collected that female, she was spent. She had released 50% of the larva. So, and normally that's not the case. That was for early March. Normally this species is gravid later. So that was an interesting result of the crazy weather that we have found. And but again, other things that can happen: dry weather patterns, like overly wet weather can affect these, the hydrologies of the river systems and water bodies. So that can definitely affect the mussels.
Right. So again, we got another one as you're answering that one. And do you have any thoughts about how pollution and increase in pollution might affect the mussels, if they are able to endure them or not?
Oh, sorry. Could you repeat that one again?
Oh, it was a question about how an increase in pollution might affect the mussels, like in terms of sediment and nutrient pollution.
Yes, like sediment, there seems to be some tolerance. There are also bioindicators. So if there's a lot of like ammonia and heavy metals, they will start dying. So if we see a die off, we know that something's wrong, and it could be chemical reasons, and knock on wood, maybe even biological. Like in the Clinch River, the pheasant shells have been decimated by what is thought to be a virus. It's called densovirus. It's only affecting that species. There's many other species in that river system, but it's killing the pheasant shells. That's just an example of, non-chemical things that can threaten mussel populations.
Can you hear me?
Yes, I can hear you.
Okay, my question actually was, yes, I understand about the pollution, but how do you test the bivalves or the mussels to see how much pollution they have endured? How do you is there a way to test inside the sample without damaging them? Is there a way you can test inside?
Yeah, if to test that, you would have to sacrifice the animal pretty much, because you need to take a look at the tissues, at the soft tissues and the hard tissues to see the contents. Yeah.
So there is no way that you can do it at this point to see how much pollution there is inside without damaging the tissues.
No. Yeah.
Okay.
And I mean, that's the same for the nutrient and microplastics. Yeah. Unfortunately, yeah, we have to sacrifice those animals for those studies. Yep.
And how many would you say that you'd have to sacrifice in order to get an actual estimate?
Well, I guess it depends on the experiment and the study. And for example, for the microplastics, we have collected about, I would say, like during the first phase, probably like 70 animals, and a similar number we're going to collect for nutrient analysis. Yeah.
And how has the Anacostia, how have the mussels affected the snakehead population? Has it decreased, increased, or is there, what's going on with that one? I'm sure you mentioned it. I'm sorry, I missed half this lecture. I deeply apologize.
You're so the main impact of the snakehead could be like because they prey on the host fish.
Okay.
But again, it would be too soon, and I don't know of any studies that have looked at that.
Okay.
But that would be the main impact, because the snakeheads don't eat bivalves. They mostly eat fish. Now, my main concern right now is blue catfish.
Catfish.
They eat a lot of mussels, and that's a known fact. Yeah. Mm-hmm.
I see.
So we did have someone in the chat who, because your daughter is sometimes being able to be seen, someone asked if you would want to introduce her.
Oh, sure. Aurora, do you want to introduce yourself?
Hello.
You're not center attention.
Oh, don't do that voice.
I'm sorry, honey. It's my cat. I apologize, but you're so beautiful.
Say thank
You're
you.
so beautiful.
Yes, her name is Aurora, or Aurora, how I would pronounce it in Spanish.
Oh, Aurora,
Yeah, so
like
yeah,
the Aurora Borealis.
yeah, and also it means dawn in Spanish. So
Yes, it does.
So we did get
So-
another question too, and I just want to quickly remind everyone to stay tuned for upcoming programs. Community Science, Capital Nature with Anna and Stella, Deep Sea Mysteries with Simone Barkley, and Natural Mysteries and Little Known Facts with Alonzo from Capital Naturalist. So those will be in the coming week. And then probably our final question for the night before we stop is just in comparison to other species or other types of bivalves, how good is the mussel filtration power, I guess, in comparison to oysters?
yeah, so oysters filter more. So one oyster can filter over 50 gallons of water a day. These mussels filter between 10 to 20 gallons of water a day. So yes, that's a comparison.
All right. I'm going to go ahead and stop sharing my screen, and we are a little past 8, so I think this is a good time to go ahead and stop for the evening. Thank you, Jorge, for joining us and giving us a wonderful presentation on mussels, and I hope to see you all at our next Naturalist Hours.
Yep.
Bye.